Hello, welcome back. We're going to be wrapping up chapter 11 with this mini lesson, taking a look at now a survey of bacteria. So for the chapter 11 material that you see in the textbook, there's a lot more information in there than what you need to be responsible for. So focus on, you have this PowerPoint, you have the lecture notes. Focus on the organisms and the information that are present in this video and on the PowerPoint. That's what you need to focus on. So don't go and memorize all the bacterial groups in chapter 11, just these selected organisms, just because they are important in various ways, and I'll try to point out that importance as we go along. All right, so we're going to first take a look at phototrophic bacteria. Phototrophic bacteria means they're photosynthetic. They have the ability to generate their own carbon, their own sugar, and then use that as their energy source. So we have a couple different groups like blue-green bacteria, used to be called cyanobacteria, or blue-green algae, but that's a misnomer because algae are eukaryotic cells. So cyanobacteria, blue-green bacteria. We have green sulfur bacteria and green non-sulfur bacteria. So if you ever go to like a pond or a creek and you see kind of the slime that's on rocks or within the kind of the edges of the water, you're most likely going to be looking at phototrophic bacteria, not necessarily algae, because this is where they like to live. They live in watery environments where they can have good sunlight, and they do a lot of photosynthesis. The next group is our gram-positive bacteria in the low GC group. The low GC is part of that classification, the analysis of nucleic acids. So organisms that have a low GC ratio, remember guanine and cytosines go together and adenines and thymines go together. So this is one of the ways that researchers have used to classify different organisms. If an organism has more A's and T's in their DNA and lower G's and C's, they belong to this group of just gram-positive bacteria. So let's take a look at who belongs in this category. The first group is that wonderful endospore producing genus called Clostridium. So these are the ones that can be pretty darn dangerous. So there's Clostridium botulinum, Clostridium tetani, so that's botulism and tetanus, and then Clostridium perfringens causes gas gangrene, which is really bad in, was pretty bad back in like before we had good hygiene, like Civil War injuries and things like that. So some of their kind of key characteristics, obviously they can form endospores, they're obligate anaerobes. We haven't really talked about aerobic organisms and anaerobes, but most people are comfortable with the idea that if you're anaerobic, you don't use oxygen. You actually die in the presence of oxygen. We're not going to talk about why that is yet. We'll see that in chapter 6. But Clostridium is an obligate anaerobe. In some of the diseases, we can see tetanus from Clostridium tetani, botulism from Clostridium botulinum, gangrene from Clostridium perfringens, severe diarrhea from Clostridium difficile. So those are some examples of the genus Clostridium. The next low GC group is called Bacillus. Now I know this is a little bit confusing because this bacilli here, this classification of the low GC includes genus of the genus name Bacilli, Streptococcus, and Staphylococcus. So it's weird to put in coccus-shaped organisms into a bacilli named grouping, but that's what the microbiologists decided. So within this larger, so this is going to be a bacilli that's not the genus Bacilli, but just a larger grouping of bacilli. We include the genus of Bacillus, the genus of Streptococcus, and the genus of Staphylococcus. So for Bacillus, some of their characteristics, they can be facultative anaerobes, which means they can live with and without oxygen. They're rods in shape. They can produce endospores, right? So they're the other endospore forming genus. They can produce diseases like anthrax, right? So the Bacillus anthracis, there's a picture of that there. And some other diseases, just general infection like vomiting, nausea, cramping, those types of things. Here's an example of Streptococcus pyogenes. So using the Streptococcus genus, this pyogenes as an example, its main physical characteristic are cocci chains. Streptococcus genus organisms can produce diseases like strep throat, scarlet fever, impetigo, and pneumonia if the bacteria get down deep in your lungs. Pneumonia is just a general term for infection of the lungs. It could be caused by various types of bacteria and viruses. And then because my head's kind of taking up this picture, I had to, this connects over here. So the last genus in this larger group of bacilli is called Staphylococcus, which is clusters, cocci and clusters. We have Staphylococcus aureus as an example. It can cause bacteremia, which is bacteria infection of your blood, food poisoning, toxic shock syndrome. So Staph aureus, this particular critter here, releases toxins that cause this, all of these different types of infections and diseases and poisonings. All right, so that was our gram-positive low GC. We had, let's see, Clostridia, Bacillus, Streptococcus, and Staphylococcus. So now we're going to take a look at some more gram-positives, but in the high GC ratio. So they have more guanine and cytosine and less adenine and thymine, and so that puts them in this classification group. So here we have one called Corynebacterium. This particular species is Corynebacterium diphtheriae, which causes the disease diphtheria, which is a pretty nasty disease if you want to read in the respiratory system. What it does is it releases toxins. It gets into your throat, your pharynx, and it produces this pseudomembrane. And basically the pseudomembrane, the toxins allow this biofilm of bacteria and cell debris to stick really tightly to your pharynx, and eventually it closes off your windpipe and you die. But we have a good vaccine for this. So this is your diphtheria part of your DTaP or your tetanus pertussis diphtheria. So we don't really see this too often in developed countries because we have a successful vaccine to block this particular infectious agent. So some of its cool characteristics is it's pleomorph. So it's mostly rod shaped, but you'll notice those rods are of all different shapes and sizes. There's no consistency between that. So that's why it's a pleomorph. It can be aerobe. So being able to use oxygen or a facultative anaerobe. It does snapping division, and it can produce what are called metachromatic granules, which are good in identifying them microscopically. But diphtheria is the primary disease that we're connecting with this particular genus of the species Corynebacterium diphtheria. A couple more in this high GC. Here's this mycobacterium. We talked a little bit about mycobacterium with the acid-fast staining back in chapter 4. So here are the two kind of most famous mycobacterium species is leprae, which causes leprosy, and tuberculosis, which causes tuberculosis. Its unique characteristics are that waxy cell wall, the mycolic acid that's incorporated within the peptidoglycan. And because of this, it doesn't stain very well, right? So since that's why they develop the acid-fast stain to be able to identify these organisms. But also they grow very slowly. So because they have to incorporate that mycolic acid into their cell walls, they can't go through binary fission very quickly. And binary fission takes normally about 1 to 3 hours for an average bacterium. The super speedy ones can do it in like 20 and 30 minutes. When we take a look at mycobacterium, their replication is more on the scale of hours to days, sometimes 10 to 24 to 48 hours to go from 1 cell to 2 cell. So when we have diseases like tuberculosis, which is, like I said, pretty widespread in developing countries, it's really hard to treat it. We have antibiotics for it, and they're susceptible to it, but antibiotics work when bacteria are actively dividing, and as they're dividing, they're incorporating the drug into their cell, and it blocks a particular metabolic pathway, like they can't make their cell wall, or they can't make their proteins, or they can't, their plasma membranes disrupted somehow. So in a normal bacterial infection, when you have replication times of 1 hour, when you are on antibiotics for like 5 to 7 days, you have a really high amount of the drug in your system, and it kills off the bacteria in those 5 to 7 days. When you're talking about mycobacterium, say like tuberculosis, if it takes 24 hours to sometimes days to replicate, to incorporate that drug into your system, you have to be on that drug for a really long time, like 6 to 12 months to make an impact on that particular bacterium that's in your body. Now, there's a lot of people that have a hard time taking their antibiotics for 5 days, let alone 6 months. So we're seeing with tuberculosis specifically, widespread antibiotic resistance. And we're going to talk about what causes that in chapter 10. But when you don't complete your antibiotic regimen, that leads to increasing in the number of cells that are resistant to that particular drug. And so we're seeing these really super drug resistant tuberculosis strains out there that we as the scientific community are having a hard time trying to get under control. So that's our mycobacterium. And one more of our high GC gram positives, throwing kind of an environmental organism in there. This is called Streptomyces. It's one of those filamentous ones that would reproduce by spores. Couple things for Streptomyces. One, they can produce a lot of our antibiotics. If you've ever taken streptomycin or heard of the antibiotic streptomycin, it comes from this particular organism. They're also ecologically important in that they play a big role in recycling organic molecules in the soil. So they're a big soil microbe. And so they're not really disease causing. Some people, if they inhale soil dust and stuff, it might cause sinus infection. But the importance of Streptomyces more is their production of antibiotics and their role in ecology. All right, so those are our gram-positive high GC. Now we're going to take a look at our gram-negative organisms. They are not classified by their ratios of guanine and cytosine. That's just for the gram-positives. Gram-negatives get more of an alphabetical kind of classification. They're called alpha, beta. What do we have? Alpha, beta, gamma, epsilon proteobacters. So let's take a look. I have one alpha proteobacter to focus on. This guy is called Rhizobium, just the genus. I'm not going to go all the way down to species for this one. But the takeaway message for Rhizobium is they're nitrogen fixers. So we can see the bacterium here, kind of on the head of a pin. So they're just showing that they're super, super small. But then on the larger scale, see these little kind of nodules growing on the roots of this plant? These little green bean looking things, not green, kind of brown bean looking things. So these are little nodules that the plant actually makes for the bacterium to live in. So it's a symbiotic relationship, mutually beneficial. The bacteria live in the nodules, and what they do is they have the ability to take N2 gas and turn it into usable nitrogen in many forms. So nitrates, nitrites, ammonias. So I didn't put those all in there. We living things like plants and animals, we cannot use nitrogen in our metabolic process. Nitrogen gas, I should say. This is in the gas form, which is about almost 80% of our atmosphere is nitrogen. So we're surrounded by nitrogen gas, but we can't use it. We need it to be converted into the usable forms like nitrates and nitrites. And the Rhizobium do that for these plants. And then we eat the plants, or the cows eat the plants and we eat the cows, or whatever your food chain path is. But the whole point is the bacteria take atmospheric nitrogen and turn it into a form that we can use, we as other living things. And so that's our beneficial relationship that we get from these Rhizobium bacteria. Without them, we wouldn't be able to do anything with nitrogen, which is super important. All right, so that was our alpha Proteobacter. I have a 1 beta Proteobacter, gram-negative beta. I selected Neisseria gonorrhea as a representative example of a beta Proteobacter. This is a causative agent of gonorrhea, some meningitis if it gets into your meninges, and pelvic inflammatory disease, especially for women. It can enter the reproductive tract and find its way into the pelvic cavity via the open fallopian tubes. So it can be pretty dangerous, life-threatening, if not causing sterility for the woman if it's not treated properly. There are diplococci, so you can see that here in the micrograph, those 2 little red circles. You can also see them in the light micrograph, so there's a diplococcus here. We can see a little cluster of 3 diplococcus right in there. Gram-negative, obviously, they live in mucous membranes of mammals, since that's one of the reasons why they are a sexually transmitted disease, because of that nature of where they live. And causing the STD of gonorrhea. So that's its name, Neisseria gonorrhea. All right, so that's our beta Proteobacter. And then I have a few examples for gamma Proteobacter. We've got 3 of them in this category. The first one is called Legionella, is the genus. The species I've selected is Legionella pneumophila, which causes Legionnaire's disease. And there's a few outbreaks of Legionnaire's disease, I think, in Disney, one of the Disney parks recently. It likes to grow in air conditioner condensation trays, right? Or big, not stagnant, but sitting water pools, so like cooling towers and things like this. And it was named Legionella because they first identified this form of pneumonia at a American Legion conference back in the 70s, like back east somewhere. People were getting sick coming from this conference, and they would go to the hospitals, and they all had the same kind of pneumonia, and they cultured the organism, and it was a brand new bacteria that they hadn't discovered before. an epidemiologist went and traced it, and it was found in the cooling tanks of their air conditioning units of the building that was housing the conference. And as they were sending out the cool air, they were running the air conditioner, it was basically just spritzing and misting Legionella pneumophila bacteria all over the crowds of people in this convention. And so a lot of people got sick. I think a few people died. It's an intracellular pathogen, which means it can actually find its way inside of our eukaryotic cells, which is unique for bacteria. Most of them just release toxins outside. And it can actually metabolize amino acids, which is also unique. Most organisms metabolize carbohydrates for their energy source. So Legionnaire's disease and a related disease called Q fever can be associated with Legionella. Okay, the next gamma proteobacter is Yersinia. The species in this case is Yersinia pestis. Historically, this is a very important organism because this is what causes the plague, the Black Death. So bubonic plague and pneumonic plague. It's a facultative anaerobe, which means it can live in the presence of oxygen, but it doesn't need it. It's coccobacillus in shape, and it has an anti-phagocytic slime layer, so it kind of helps to keep it in the body and not succumb to phagocytosis. Bubonic plague, so you might be wondering, well, where did the name bubonic come from? Well, it gets into your lymph nodes. So this swollen area right here is called a bubo, right? So it's right here in your cervical lymph nodes, but it could also be in like the inguinal lymph nodes of the groin. So if you take in 233 lymphatic system, you know what I'm talking about. So they get swollen and big, and this is in what that's called a bubo. So bubonic plague, you'd get that as a result of getting this infection. And then pneumonic plague goes into your lungs, which is way more deadly than the bubonic plague. And it's also called the Black Death, right? Because look what happens to tissues sometimes is the result of this organism kind of breaks down tissues, breaks down cells, blocks circulation to your extremities, and you get necrosis of your tissues. And so this person's going to lose all of those fingertips. There's no way that tissue is going to come back. Unfortunately, that's pretty severe. And so this is the plague that killed a third of the European population back in the 1300s, I want to say, 11 to 1600s when that was kind of circulating. It is a vector transmitted disease. It lives in fleas, which are arthropods, which live on rats and mice, right? So if you remember your history, if you learned about the Black Death, people didn't have very good hygiene back then. They lived with rats and fleas was a common occurrence. And then the disease would go into the flea would go from the mouse to the human, it would feed on the human, and that's how it would transmit the bacteria. And there was no hygiene, there was no treatment, so you survived if your immune system could fight it off. There's still Black Plague around. It's usually in the west and southwest. There's a case of a guy got it from his dog, I think, or from a flea from his dog in Oregon not too long ago. But we have antibiotics now, so it's not as dangerous as it was back before we had antibiotic treatment. Okay, and the last of our gamma proteobacter is an organism called Klebsiella is the genus. I'm picking Klebsiella pneumonia as the representative species. Again, gram-negative. This has a really good capsule. We actually, in an in-person lab, we stain Klebsiella pneumonia with our capsule stain, and you can see these big, thick, complex polysaccharide capsules. They're aerobic in their metabolic pathways. They cause pneumonia, as the name implies, Klebsiella pneumonia, and they have they produce what's called CRE infection. CRE stands for organisms, bacteria that have found resistance to a particular type of antibiotic called carbapenem. So carbapenem resistant Enterobacteriaceae infection, so CRE for short. And Klebsiella is a type of bacteria that have acquired those resistant genes to not be killed by this carbapenem, which is a last resort antibiotic for these particular types of infections, which is really scary. All right, our last proteobacter group is Epsilonproteobacteria. And I have two examples for this one on the same slide because they're both dealing with the digestive system. So we have Helicobacter pylori and Campylobacter jejuni. I didn't put the species names on there, so we'll just stick with the genus. Helicobacter causes stomach ulcers, and that's these guys right here. This is your Helicobacter, kind of a little spirally. And then our Campylobacter, and here's the ulcer. kind of the open wound in the lining of your stomach and your digestive tract. And this is our Campylobacter right here. It's the most common form of food kind of gastritis when you eat. It's usually associated with undercooked poultry like chicken and stuff. And so gastritis is very common for this Campylobacter jejuni organism. All right. Okay, last group that's not it's gram-negative, but it's not in the Proteobacter groups. It's spirochetes. So just real quick, we're going to talk about spirochetes. We have Treponema and Borrelia. So Treponema pallidum causes syphilis, and Borrelia burgdorferi, I think is its species name, causes Lyme disease. So syphilis is a sexually transmitted disease. It can be treated with antibiotics, but there's some antibiotic resistant strains out there. It only lives in humans. Humans are the only host for that particular organism. And if it's not treated, it can lead to neurosyphilis, which is a permanent denigration of your neural functions. And then Lyme disease, here's kind of that telltale bullseye rash that might show up, is a vector-borne illness. The bacteria live in ticks. Usually on the East Coast, but there's a few of those deer ticks that can transmit Lyme disease here on the West Coast as well. So you want to be super careful if you go out in places where ticks are formed. You always want to do a tick check when you come back in and remove them if you have them as quickly as possible. I think the understanding is it takes about 24 hours. You have to have a tick kind of feeding on you for 24 hours to increase the likelihood that it's going to spread the Borrelia into your bloodstream. All right, that's it. So that was our survey of chapter 11, dealing with the bacteria, the different gram-positive groups, the different gram-negative groups. Again, don't memorize the whole chapter of 11. There's a lot of data tables in there. There's a lot of information. If you just focus on what I've presented in these slides, then you'll be good for assessment opportunities that are coming up for the tests and things.